Hello everyone, welcome to lecture 11, and today's lecture is going to be about macroecology. Okay, let me share my screen. Okay. So first, what is macroecology? Well, so this is one definition by Jim Brown, who is one of the founder of macroecology. And so in his 1995 paper, he mentioned that macroecology is a way of studying relationships between organisms and their environment that involves characterizing and explaining statistical patterns of abundance, distribution, and diversity. So here, this is a way of studying. So it's more about the methodological side of ecological research here. And so why they use this macro as a prefix? Well, they mentioned that there are two reasons for this. So first, because in order to characterize patterns in the statistic distribution of variables among individuals or populations or species, it is usually necessary to work with relatively large spatial and temporal scales so that we can obtain sufficiently large sample size or data sets. And the second reason is to investigate the implications of important advances in other disciplines, such as biogeography, paleobiology, systematics, and the earth science. It is necessary to expand the scales of ecological research. And so in one of the assignment reading, by Brian McGill, the what, how, and why of doing macroecology, and he reviewed two major views of what is macroecology, and he also gives his own view of what is macroecology. So the first view is macroecology is equal to the ecology of large scales of but then this is sort of vague because how large is large enough? And we also know that when we talk about scales, we have the extent and the grain. So we talk about large in extent or large in grain. And also when we talk about scales, what kind of dimensions we talk about? And here in his paper, well, in general too, we can think about the dimension of space, time, and taxonomy. So looking at space, so we have less than 1 square meter to 1 square kilometer, then 10,000 square kilometer, and even larger. So this is the spatial dimension. And what about time? So we can talk about 1 hour, 1 day, 1 year, 10 year, a million year. And we know that the Earth's history now is more than 4.5 billion years. So this is the dimension of time. But we also, when we look at macroecology, we're studying species. Okay, so then that's another dimension that's about taxonomy. So we can talk about 1 species, 100 species, or 10,000 species. And so far, the sort of expected species on Earth, now I think it's more than 10 million species there. And most of them we don't know yet. And when we look at this 3 dimensions, and this X mark here is the main large in 0 dimension, and this corner is most of the previous traditional ecological studies are about. So we look at relatively small spatial area across relatively short time period, and we look at either population or a few hand of species. And here, macroecology, we're extending the scales of different dimensions. Like, so this light gray balls, they are just large in one dimension. So here, take the space dimension, for example. Uh, so here, it's a large space, but short time and potentially only about a couple of species. So that's one example of a large in one dimension. And then the gray balls, they are large in two dimensions. So here, for example, this ball is large in both space and time, but small in number of species. And this one, it's large in space and large of number of species, but short in time. And this one, we are talking about um, lots of species across over a long time period, but only at a small area. And this black bowl is large in three dimensions. So now we're talking about lots of species across a long time period, across large area. So it's going to be large in three dimensions. And so this is the three very common dimensions when we talk about scales. And macroecology does not require to be big simultaneously in all dimensions. It can be, say, big in number of species and a large area. So that's basically spatial patterns of, say, species diversity. Put that as an example. And also, when we talk about large, how large is enough? Well, this needs to be defined relative to the organisms involved. And in this McGill 2019 paper, Brian mentioned using the notion of dozens as sort of a threshold. So dozens of generations of time, then it's going to be potentially large enough. And then for space, then we can think about maybe dozens of population and the area they're occupying. So that's going to be the space dimension. And for species, if we have more than a dozen of species, then potentially this can be, we call large enough to the taxonomic dimension there. And now let's talk about the second view. So that is view macroecology as a statistical, emergent, and non-reductionist approach. So this quote from Smith et al. In 2008 is I think it tells it all. So macroecology is a big picture statistic approach to the study of ecology by focusing on broadly occurring patterns and processes operating at large spatial and temporal scales and ignoring localized and fine-scaled details. So ignoring details, macroecology aims to uncover general mechanisms operating at organisms, population, and ecosystem levels of organizations. So, so there's one thing I wanted to highlight: ignoring localized and fine-scale details, because so in the reductionist view, adequate modeling of the component should always reproduce all emergent phenomena. So think about the traditional ecology. So that covers most of the topic so far. For example, we talk about species interactions like competition or resource consumer relationship there. We talk about relatively small scale, localized, fine scale details. So that's the thing we talk about. But when we look at a macroecology, we're actually scaling it up to look at the large scales, and that scale potentially the smaller fine scale details are just not going to be important, while environment potentially getting to be much more important in terms of organizing of ecosystems or populations. So that's sort of the second view of macroecology. And then why, you'd ask why this is going to be an emergent and non-reductionist approach. Well, the main problem here is the so-called transmutation problem. And we know that scaling up is a challenge because of adjacent inequality, which we have talked about it multiple times across this semester. And put it in a simple way, it's just saying that the average of a function is not the function of the average. So for example, this fx then bar, so that the bar represents the average, okay? So f is a function, and the function of xi, say we have 6 of that, for example here, the rainfall and the biomass. So x-axis has the rainfall, y-axis has the biomass. We pick 6 points, so 6x values, x1, 2, 3, 4, 5, 6, okay? So then the corresponding y value is going to be fxi, okay? So here, fx1, fx2, fx3, fx4, fx5, and fx6. And then the bar of this fx, so which is the average of the function, is just take the average of the 6 values, then that's going to be this horizontal dotted line, okay? So this is fx bar. But this is not going to equal to fxi bar. So what does this mean is that we take the average of x1 to x6. So this vertical dashed line represent xi bar, okay? So that's the average of x1 to x6. And then what is fxi bar? It's going to be here. This is the horizontal dashed line. And here we can see that the horizontal dashed line is above the dotted line, which means that the fx bar is less than fxi bar. So, and here in this example, the error is about 10.9%. Okay, so that's briefly the adjacence inequality, and that's a transmutation problem. And put it in real data. So here, x-axis is annual precipitation, and the y-axis is annual productivity, and each dashed line is one site. So one site over time. So we have variation in annual precipitation as well as variation in productivity. And within each site, we can see we have a strong positive relationship and no relationship and even negative relationship with high precipitation. Well, why this negative relationship? Well, one potential reason is that, with high precipitation, it's going to wash away the nutrient. And now the plants are not limited by water, but instead they are limited by other things like the nutrient of the system. And also with too much raining, then they potentially can suffer from flooding. Like their roots are underwater, so it's going to be a trouble for them. And but if we look at all the sites together, and it is clear that we get this positive nonlinear relationship between precipitation and annual productivity, which none of the individual sites tells us this. So even though we study each side, uh, and if we use this reductionist approach, we're not going to produce this emergent pattern here. So this justified the importance of macroecological study. Okay, and then we move on to the third view put by McGill. And he argued that macroecology is the study and the aggregate level of aggregate ecological entities made up of large number of particles in the pursuit of generality. So here, one example is that this different small circles, they are just different particles, and we aggregate them together. So then this is going to be the black large circle. So that's aggregated ecological entity. And we're going to study that. So this telescope is kind of represented as the macroecological research. So we're looking at this aggregated level of this aggregated ecological entities. And that's so focus on aggregated unit and aggregated levels instead of just say scales, like the first view, but it's also sort of combine the first view, the first two views to form this like the third view here. So that's what we learned from the reading paper. So the different views of what is macroecology. And now we are going to move on to talk about different macroecological laws. And here I put laws in quote simply because they're just patterns and hypotheses, and not every hypothesis are uncontroversially supported. So, and here we're going to talk about the four different groups. So that's species richness and species range size, species body size, as well as species abundance. Okay, let's first look at the species richness. And one common pattern is longitudinal gradients in species richness. And here is a map of species richness of vertebrates across the world. And the warmer the color, so from red to dark red, that means more species. And with a cooler color like blue, deep blue, they are like represent smaller species richness. Okay, and now the pattern is quite clear that in tropic areas, so this is Arizona, sorry, so this is the Amazon or Andes area, and this is the rainforest in Africa, and this is the Southeast Asian area. We can see the tropic areas are much, the color are much warmer than the polar areas, which means that we have more species in the tropic area. And so this pattern of increasing species diversity from the poles to the tropics is remarkably consistent across different taxa groups, either marine or terrestrial groups, like from plants, fish, mammal, bird, and many invertebrate groups too. Of course, there are also exceptions can be found. But by and large, this pattern is very consistent, and this pattern also persists across much of both modern and deep time. So which means in the past and also now, this pattern holds. Okay, so then we have this remarkably consistent pattern, but then we ask why, why we have this longitudinal gradient in species richness. Well, over the past decades or century, dozens of hypotheses have been proposed. Well, if we think about species richness And essentially, we are looking at speciation and extinction rate, and we can ignore the dispersal and the immigration at this moment. And so the speciation and extinction, both of them determine like the species richness of an area, okay? And we can group this different hypotheses into four large categories. So first, that's the climate. And because of the shape and the position of the Earth relative to the sun, and that tropic area is going to receive more solar energy. And it's going to also have a lower seasonality. So this one need to high productivity. And with high productivity, we're going to have more food to support more species through food web. So this is going to be also transferred to higher species richness compared with high latitude area where it has less energy and lower productivity. So that's sort of the climate group of hypotheses. And then the second group is about time, um, historic hypothesis. And in tropic area, because it has been less perturbed by the climatic event, such as glaciers. We know, like more than 10,000 years ago, like half of the North American are covered by ice. So, but not in tropic area. And tropic area, because they are less perturbed by this climatic event, they have more time to accumulate species over time. So therefore, this can explain um, we have why we have more species in tropic area. And then the third group of hypotheses is about geographic. So because the tropic area has large area, and also this area are more heterogeneous. So this can increase speciation rate. Um, and therefore it's going to be able to have more species richness. And finally, we can talk about this mid-domain effect. So by extension, it's like, if we have bounded geographic domain, and if we just randomly shuffling species range, with with their latitude and range size to be fixed, then we can randomly put that species within the domain. Then this just by chance is going to produce a mid-domain peak of species richness. And we can use null models to test whether the observed pattern of magnitudinal gradient of species richness, whether that can be produced by chance unknown using null models. I think we briefly talked about null models in the lecture of network. So the key idea is just to keep the statistics or the property that we are interested in, and to keep all other things the same, but just vary the measures that we are interested in. And if we do this randomization, say 1,000 times, then we're going to produce a null distribution of the measurement. And then we can compare what we observed with this null distribution. And if, say, the observed pattern, the observed values is far away from the null distribution, then we can safely say, no, the observed pattern cannot be produced by chance. Okay, so that's sort of the four different groups of hypotheses to explain longitudinal gradient of species richness, but they're not mutually exclusive. And also, this different group of hypotheses, they can work together, they can work simultaneously, potentially to produce this longitudinal gradient of species richness. And here we focus on longitudinal gradient, but also we can find a similar pattern of elevational gradients too. And so from the bottom of the mountain to the top of the mountain, we can potentially, observe the decreasing species richness too. And while the hypothesis, some of them can be explained, using similar hypotheses to explain the longitudinal gradient, but also the other hypothesis to explain elevational gradient. And we're not going to cover that here. Okay, now let's move on to talk about species range size. First, we'd be interested to know what's the common range size distributions. And here, at a small spatial scale, and this bimodal distribution is not uncommon. Okay, so bimodal means we have one peak at the lower end, lower range size end, and one peak at the higher range size end. So the y-axis is the number of species. So this means we have lots of species that are widely distributed across the small area. So again, we talk about small spatial scale. So we talk about small areas, okay? And we also have lots of species, they only observed in a small part of the small area. So potentially, this and, more common, the range size cover the whole small area. The sort of more widely distributed, but this rare species, or small area species, they sort of probably be specialist. They adapted to a particular environment. And so this is quite interesting to have this bimodal distribution at small spatial scale. But when we look at a large spatial scale, the pattern can be different. And here is an example, like if we take a log transfer of the range size, we sort of get a close to normal distribution pattern here. Again, the y-axis is the number of species, and we can see, the most of the species in the middle, they have a sort of mediate range size. Few very common, like say worldwide distributed species, and also relatively few species that are only observed in a tiny area. So that's sort of the how the range size is distributed across different scales. But we can also talk about spatial variation in range size. And one particular pattern here we call rampant row. So that's the tendency of species living at high latitude to have a larger range size. And here is an example of bird species across the New World. And the x-axis is the latitude, and close to zero, that's the tropic area. And then the y-axis is the mean log range size. And we can see this this shape of V shape, here is more clear with the different masses. So A and B, they're just different masses. So clearly that the range size is smaller in tropic area compared with high latitude area. And so why, again, what causes this pattern? Well, one potential reason is that in tropic area, it's going to be the competition among species is going to be more strong. So therefore, the species are going to have a narrow niche. So because this competition is so strong to force them to be specialized and to have a small range size and to have a narrow niche. Therefore, this kind of can result this V-shape, and this particular um, sort of pattern can also be contribute to the longitudinal gradient of species richness. Okay, because the species in the tropic area, they have a smaller range size and a narrow niche. Potentially, we can pack more species in the tropic area. Okay, and another potential reason is that in tropic area, because higher species richness means more species interactions, and therefore species are potentially more controlled or affected by predators or parasites or pathogens to make them the range size to be smaller than area in high altitude and high latitudes. Okay, so that's spatial variation in range size. And what about body size? So body size distribution, one common way to measure body size is just to measure the body mass. And this can be a universal measure because every species of every individual has body mass. And we can take a measure of that and compare that. And the frequency distribution of body mass, so again, taking a word birds as example, if we're using the raw body mass, it's going to be a highly skewed. So this is a skew to the right, skew to the right distribution. So the x-axis is the body mass, and the unit is a gram. Okay, so 80 means 80 kilogram here. So we can see very few species, very few birds is more than 80 kilogram. Uh, but a lot of them actually, thousands of them to be less than, excuse me, less than 10 kilogram here. So highly skewed. And if we take a log10 transfer of this body mass, then we sort of get to a close to normal, but not quite there yet, normal distribution here. And there's actually both biological and statistical reasons that this log10 transformation is quite common in biological measures such as body size or seed mass, um, is extraction, uh, yeah, some sort of examples like this. We can talk more in the lecture on Thursday if you're interested. Okay, so that's the body size distribution. And we can also talk about the spatial variation in body mass. And here, one particular hypothesis is Bergman's rule, so which is the tendency for a positive association between the body mass of a species in a monophyletic, well, you can ignore this, higher taxon, and the latitude inhabited by those species. So again, take the word, take the new word bird species as example, and here x-axis is latitude and y-axis is the average body mass. And we can see that this is a U shape here. So which means at tropic area, the body mass is smaller than species and high latitude, high latitude areas. Okay, so then again, what's the mechanism behind of this pattern or this hypothesis? Well, one common mechanism or hypothesis is that it has something related to heat conservation. like in cold area, if you have a large body, then you're going to have a lower surface area to volume ratio. So this will conserve uh, conserve um, the heat of the body. And on the outside, in warm area, like in tropic area, then if you are smaller, then you have higher space area to volume ratio. So that potentially will allow you to improve the heat dissipation and to be better adapted to hot weathers or hot areas. So that's one very common hypothesis. But also, we can think about the, starvation resistance. Like if in a cold area you have less food, right? And then you probably don't want to be starved. So therefore, if you have a large body mass, you potentially will be more starvation resistant. But also there are other hypotheses regarding the evolutionary history or dispersal ability, things like that. And if you're interested, I'm sure you can find papers about that. And we are not going to cover that here. And but also for the evidence, what's evidence for the Bergman's rule? It's actually quite mixed. Like one recent example is that by this paper, they didn't find a general relationship between body mass and temperature in endothermic species like bird and mammals. And they look at the hundreds of species there. Okay, so now let's move on to species abundance distribution here. And species abundance distribution characterizes the distribution of abundances of all species within a temporal or ecological community. And there are two primary ways to visualize species abundance distribution. And the first one is just to take the histogram of abundance. And here, x-axis are just different groups of abundance, okay? So number of individuals. So, for example, the first bar is like 1 to 2, then 2 to 3, then etc. etc. So we've been species with different abundance there. And then the y-axis is just the number of species that the abundance fall within this bin, okay? And we have this, again, highly skewed to the right pattern, and which we call the Hollow curve, And so what this really means is that rare species are common because, so here, this highest bar, they have the lowest number of individuals, so they're rare. So rare species are common, and common species, which means the abundance are high, so we have more number of individuals, but they are rare. So because you only have, like in this example, less than 10 species to have high abundance. So common species are rare. And this pattern, so this rare species are common and the common species are rare, are generally true in most ecological communities, no matter what scales you look at, like small region or large region, this pattern holds across spatial scales. Okay, so then the downsides of this way of visualize species abundance distribution is that we need to group species, and by grouping species, we potentially lost some information there. And also we need to think about what's the size of the bin. Like here, we just 1 to 2 or 1 to 3 or 2 to 3, then we can potentially put it as like 1 to 3, and the bin size of 1 or the bin size to be 2, the bin size to be 5. So different size of bins can affect the results too. And so how to choose the bin size is another sort of a arbitrary thing we need to do. So that leads to the second way to visualize species abundance or distribution, and that is the rank abundance distribution. And here we rank the species by their abundance, and with 1 to be the most abundant species. And the y-axis here is their abundance. It can be scaled to be relative abundance. And so this rank abundance distribution can tell us species richness. So that is how many rank here. So for example, if this is 270, then we know, oh, this system or this community has 270 species. So this can tell us the species richness, but it can also tell us the species evenness. So we can see that, so this is like, a curve. And if say we have a horizontal line here, all the dots fall within the horizontal line, which means all species have the same abundance, which is not going to be happen in natural community. Okay, just a hypothetical example there. So that's going to be high. It's an even distributed community, and so therefore this curve, how deep, how steep this curve is, can tell us about evenness. That is how how even the species abundance are. Okay, so these are two primary ways to visualize species abundance distributions. And then we can talk about one important hypothesis, that's abundance center hypothesis, which states that species density should be highest in the center of species range. So here is one example taken from Tad's paper. And this is a mice, and you can see the blue area is its distribution range, and this red square is represent the center of its spatial distribution range. And then we can have different dampers and different communities, and we can calculate the distance from the damper area to the center. So which is going to be the x-axis here in B and C. And the difference is here, B is a geographic distance, just the distance, while the C is the environment distance, okay, from the center. And we can also, and this damping site, if we have the same area across different damper area, then that's actually, we can calculate the species density. It's going to be proportional to the abundance too. So if we plot this abundance against the distance, and if we have a negative relationship, which means the closer you have higher abundance, it's going to give us support about this abundant center hypothesis. And I think in this paper, in Tad's paper, he didn't find strong support for this for this hypothesis. And recently, last year, he synthesized all the research about this hypothesis and find again the evidence for this hypothesis is mixed. And with most of them on, most of them just don't support this abundance center hypothesis. And if you're interested in, I put the link of this paper here. If you click on this red text, you should be able to find the paper and read about all the hypotheses that Tad and his co-authors put together to explain why the evidence is mixed and on what in the future we can do to further improve our understanding about abundant center hypothesis. Okay, I think that's all the sort of macroecological laws, we the common laws or common patterns in macroecology, and and that that's all the things I have to show. And by the end, I wanted to post this question here, and also I want to highlight the importance of macroecology in an era of a rapid change. So from the last lecture, we know that the environment is changing rapidly. So things like climate change, land use change, and biological invasions, how do they individually or together interactively affect this different macroecological patterns such as species range size, abundance, body size, and diversity is going to be a really important question, and because ultimately that's going to affect the human society. Okay, so take an example of body size. Um, and if, say, Bergman's throw is true, then we'd expect with future warming, climate warming, then we'd expect the species body size to be getting smaller and smaller because they need to adapt to the climate, and if they're smaller, they probably will be better adapted to the warm climate. Um, Given the hypothesis we talk about regarding heat conservation or adapt to hot environment. And if this is true, then then things can be getting worse, because if we have smaller body size, then potentially, that means we human get less food. And if this holds um, for for across species, then also, all the mammal species, they're going to have less food. Therefore, it's going to have consequences. We talk about the networks, we talk about the food webs in previous lectures, so we know things are connected. Okay, one thing's change can result of a chain of changes. Uh, they are like a trophic cascade impacts there. And but on the other hand, so that's a climate change, but also what about land use change, like urbanization? So urbanization, how urbanization is going to affect the body size? Well, think about, like, in urban area, potentially it's going to be warmer than the rural area. So suppose the species in urban area is going to be smaller, but on the other hand, urban area just provide more food source, right? Like a human just waste so much food, like in the trash cans, and that means more food for animals who are adapted to urban area. So therefore, like, even though it's warmer than rural area, but they have more food to eat, then potentially they can have even larger body mass. So we can see now, how this different aspects of environmental change, like climate and land use change, can interact together to affect this different ecological, macroecological patterns. Like this is true for body size, but also think about range size. Like, in this background image, it's a highly fragmented deforestation area. So this is like a rainforest jungle. We know lots of species, they are specialists, and they call this habitat home and the only home for them. Without this habitat, uh, and they're going to, if the area of this habitat is going to shrink, then the range size is going to shrink undoubtedly. And if it's going to be small, made too small, it's not going to be enough for them to support a population size that to be able to sustain. So therefore, extinction is unavoidable if the habitat is too small for them. And not just land use change, but also, biological invasions can potentially affect the species range size. So we talk about that in, I think it's a lecture of disease. So the invasion of virus or pathogens can potentially wipe species, out of their occupying the range. So that will reduce their species range size. And also, this is going to affect the human society ultimately. So talking about like invasions of virus, like, we're still deep in this pandemic. And whether in the future with rapid environmental change, whether this invasion of virus to be more frequent and to be more impactful to human society, we don't know. But like also this macroecology about the virus, about pathogens, about parasites. So that's all important in research. So that also highlights the importance of macroecology in an era of rapid change. Okay, I think that will conclude this lecture, and I hope this lecture um, and also all the lectures across this semester will allow all of us to be more informed citizens, no matter which career you choose. If you choose to be a doctor, or to be an ecologist, or to be any any career you choose, I hope this semester, this course can allow us to judge things critically, to think about consequences of our daily actives and our actions, and with the yeah, I really hope this can be helpful for us to be more informed in terms of to be more aware of the environmental challenges or ecological challenges we are facing, but also the challenge means lots of opportunities for us to understand this challenge and to find the solutions. I think together we can do this. We can create a sustainable future for the next generation and for the generations of the next generations. Okay, thank you so much, and I think we can talk more in the Thursday's lecture. All right, thank you. I will stop recording.